This would be the last chapter of of biochemistry, which we'll be discussing in our today's class. So, in this chapter, we'll be talking about the various catabolic pathways. Okay, whether we talk about using carbohydrates, because we know that body can use not only glucose as a source of energy, body can also use fats, proteins as the source of energy. Okay, so we all know that glucose is used as a source of energy. We know that, right? It's the preferred source of energy in our body, right? But what if glucose is not available? Okay, so in absence of carbohydrates, what does our body do? Okay, our body uses fats first as a source of energy, and if proteins are, overabundant in large quantities, okay, in the body, then body can use proteins as a source of energy. We call them non-carbohydrate resources. So we're going to talk about all these here. Okay, we got to grab a pen, right? So we will start with the how glucose is used as a preferred source of energy. We'll talk about the glycolysis in detail. That is the main attraction of this chapter. Okay, so we'll talk about how, see, if we talk about digestive system, we know that how monosaccharides are produced, right? We spoke about all that in the digestive system, how we had the polysaccharides and then we had the amylases acting on them, the amylases in the mouth, right, the salivary gland, and then we had the pancreatic amylases, and then we had the brush border enzymes, right? And after the action of brush border enzymes, right, like your lactases, amylases, proteases, right? Finally, you will have the monosaccharides, right? So then in this chapter, we'll talk about how your glucose is used as a source of energy. Okay, that's the first part of this chapter. Later on, we also know that, as I just now mentioned, that we can also use fats as a source of energy. Okay, and we also spoke about in digestive system in PHSF how you have the lipase present in the mouth, the lingual lipase, and the lipase present in the pancreas, the pancreatic lipase, right? These two act on the lipids and convert them into glycerol and the fatty acids. Remember, it's not the bile which digests the fats, it's the lipase. Bile simply emulsifies the fat. We discussed that in digestive system. It does not digest the fat, it simply causes emulsification of the fats. Okay, so we're going to talk about the beta oxidation of fatty acids. Basically, when there is not enough glucose present, okay, to fuel our cells, okay, our cells, okay, how we can use large amount of fats to produce the ATPs. Okay, we call it the beta oxidation of fatty acid, which is pretty much faster, okay, than anything else. And then at the end, this chapter, because this chapter has three parts, right? This is the first part, wherein carbohydrates as a source of energy. Second part will talk about how lipids are used as a source of energy. And the third part will talk about how proteins can also be used as a source of energy. About the protein digestion in PHSF, we spoke about proteins are not digested in mouth at all. Protein digestion begins in the stomach with the hydrochloric acid converting the pepsinogen into the pepsin, right? And it's the pepsin which will break up the proteins. And finally, when proteins will reach to the small intestines, and then we have the brush border enzymes as well as the pancreatic enzymes, which will convert these into amino acids or dipeptides or tripeptides, which are absorbed. And then how these amino acids can be used as the source as a source of energy to produce the ATP. But when proteins are overabundant, okay, and when carbohydrates and fats are not available in the body, okay, at that time we use the amino acids. They are oxidized for the energy. Okay, this is all about that, and you will see that we will talk about this is like a main cycle. Okay, so in this cycle we'll talk about how carbohydrates, we know how carbohydrates are converted into monosaccharides, then how glucose is converted into the pyruvate, right? Stages of which respiration? Cellular respiration. If you remember last class also, I told you that cellular respiration involves four different phases. One was the glycolysis, right, which takes place in the cytoplasm. Second was the formation of acetyl CoA. Okay. And then we have the third phase, which we already discussed about the TCA cycle. Okay. And then the fourth phase was this is the fourth phase, oxidative phosphorylation. Okay. We discussed the electron transport chain. We already discussed these two, we discussed previously, right? And then finally, how ATPs are produced. Okay. And then but you will see that, okay, you will, as we will move on towards the end of the chapter, you will see that, as I said, that we'll use lipids which are converted into glycerol. They can also join the glycolysis. They can also, you know, act as an intermediates and produce the ATPs, enter the glycolytic pathway. Or even fatty acids, okay, their beta oxidation can form the acetyl CoA, and that acetyl CoA can be later on used by the TCA cycle or the oxidative phosphorylation to produce the ATP. So you can see them, you know, they can intermediately join in between, right? So other than that, you will also see that how proteins join. Okay, we'll discuss. So this will not be very clear at this moment, but as we will move towards the end of the chapter, so you will see that how proteins enter into the pathway, how they are used to generate the energy, right? So we're going to discuss about all these things. So so not necessarily always carbohydrates can be used as a source of energy. We can use fat as well as the proteins, which we will see in this chapter, right? But at the end, if the oxygen is available, then it is the oxidative phosphorylation and the Krebs cycle, which will produce a lot of ATPs. Are we okay about it? So did you all understand what this chapter is all about? This is just the introduction about the chapter. Yeah. So in this is like an overview of the chapter, right? So then we have, okay, then in this diagram, it's simply telling you the same thing, that how complex molecules like your starch, proteins, lipids, right, how they are catabolized, okay, and how each bond is broken in the food and they are used to produce the ATPs with the release of some energy, okay. And once we have the simple molecules, they can be used as the source of energy, okay, while producing the ATPs. And these simpler biomolecules like fatty acids or amino acids or glucose can also be used in the anabolic reactions, right? Basically, that chapter we're not going to discuss. That was the last chapter, right? How the body building things, they can also be used to produce, for example, small amino acids can be used to produce larger proteins where the heat is released. It's simply explaining you about the catabolism and the anabolism here, which we already discussed in the previous chapter. Okay, we start with the glycolysis. Okay, so can anyone tell me where does glycolysis take place?
The cytoplasm.
In the cytoplasm, absolutely right. So what is glycolysis and how exactly it takes place is basically the things which we are going to talk about here, okay. So all the reaction steps of the glycolysis takes place in the cytoplasm, okay. Next question, where does it take place in the body? Can you name any specific cell where the glycolysis can occur? Do you think there's any specific cell
Muscle
where?
cell.
Muscle cell, okay. Only muscle cell, okay.
All cells.
Yes, very good. Absolutely. All cells in your body, all tissues in your body, right, are capable of doing the glycolysis. Okay, so this is the only pathway which you will see in the entire biochemistry. This is the only pathway which basically takes place in every kind of the cell. Otherwise, you will see that one pathway will take place only in this one, only in that one, only in kidney, only in liver, only in brain. But this is like, this takes place everywhere in the body. And for the RBCs, this is the only source of energy, because RBCs do not have mitochondria, they cannot do the aerobic respiration. So for the RBCs, this is the only source of what? Only source of ATP generation. Okay, rest it takes place in every part of the body. Okay, so before we discuss about the glycolysis, how many reactions are there, let's try to understand what does the word glycolysis mean. Okay, glycolysis. Okay, glyco is basically gly, gly, right, is something which is very sweet, like glucose. Okay, and lysis, lyss, you know that lysis always means splitting, right? Lysis of the cell. Okay, so lysis means splitting, so glycolysis means we are splitting what? We are splitting the glucose. That is our main aim. Splitting of glucose is called as the glycolysis. We have 10 reactions. You have to remember this as well. We have 10 reactions or 10 chemical steps happening over here, where the glucose will be converted into two pyruvic acid molecules. Remember, this will help you. This 2 is very important. This will help us in the ATP generation at the end. Okay, so remember that one molecule of glucose is converted into two molecules of pyruvate, a very important point. So if one ATP is produced at the end, you have to multiply it with the 2. That is very, very important. Okay, is it clear? So what are we aiming for? We are aiming for what? What do we want at the end of the glycolysis? What should be? As I said that before, you discuss any pathway, ask a question to yourself: why I'm learning this? What I'm going to get after this? We are we are simply trying to convert the glucose, which is a preferred source of energy, into the pyruvic acid. That is our aim, so that we can take that pyruvic acid into the next second stage of the cellular respiration, where we will form the acetyl CoA, right? And if you remember last chapter, we said that acetyl CoA combines with the oxaloacetate. Remember TCA cycle, the first step of TCA cycle, right? Acetyl CoA combines with the oxaloacetate to form the 6 citrate, right? Which was a 6 carbon compound, right? So that is why the aim is so that we can split the glucose, form the pyruvic acid. From the pyruvic acid, we can form the acetyl CoA, and that acetyl CoA we can use it in the TCA cycle, which we already learned previously, how it combines then with the oxaloacetate, right? And then, if you remember in the last class, we spoke about how then NADH, FADH2 are produced, which are taken to the what oxidative phosphorylation electron transport chain to produce many ATPs. So that is our aim. That is why we are doing glycolysis, right? But do we generate any ATPs in glycolysis? Do we? That is a question which you need to ask yourself. Yes, we do. Even though it's not like your oxidative phosphorylation, the way we discussed it there. Okay, there we had large amount of ATP is produced. Okay, but here you will see that even though the yield is very less, but it is something which is very, very important, because of the glycolysis we will get pyruvate. Because of the pyruvate we will get acetyl CoA, right? So eventually that acetyl CoA will be taken to the Krebs cycle. So that is why it's very, very important. As I told you that before we start any pathway, you should be very clear about that why I'm studying this, what I'm going to get after this, out of this. Okay, so what is the net yield of the glycolysis? We'll see that we will produce 2 ATPs at the end. We'll actually produce 4. You'll see that at the end we'll be using 2 of them. So the net gain will be the 2 ATPs. Okay, so you can say in terms of energy efficiency, it's not the good pathway. It's not very efficient because it just produces 2 ATPs per glucose molecule, right? And you will see that it will also produce some NADH as well. Okay, but still most of its energy is locked up. Okay, is locked up in this pyruvate. So this pyruvate is going to fetch us a lot by converting it into acetyl CoA. If you see here, this pyruvate is going to get converted into, let me erase all this. This pyruvate is going to get converted into acetyl CoA, and this acetyl CoA is going to fetch a lot of energy when it enters into the citric acid cycle, and then when NADH, FADH2 are produced, which enters into the oxidative phosphorylation. So this is the basic aim of glycolysis, right? Even though the energy yield is less, but we know what we are producing. We know we are producing the acetyl CoA, or we are producing the pyruvate, where we still have a lot of energy stored in the bonds of the pyruvic acid or the pyruvate. So is the overview clear to everyone before we actually start discussing what glycolysis is and what are the various steps of it? Any questions till now? You all understood why we are doing glycolysis, right? Very good. Okay, Alhamdulillah. Okay. Now, moving on further, let's discuss the glycolysis in detail. Okay, before we start discussing glycolysis, as I said, that what are we aiming towards it? At the end, you will see that we will produce two ATPs. You will see that we will produce the NADH, and you will see that we will produce two molecules of pyruvic acid. You should be very, very clear about it, because these are the questions which are usually asked in the examination. Okay, and you will see that here, one more important point here, we don't have any electron transport chain here, no proton pumping and all here. Simply how the ATPs are produced here, we will do the substrate level phosphorylation over here, right? So substrate will be phosphorylized, and that's how the ATPs will be produced. This is also very important. Okay, the substrate level phosphorylation takes place in the glycolysis. There's no proton pumping and all that here. Are we okay? Should we start discussing it? Any questions? Okay. Okay. So this is basically glycolysis here. Okay. First, we will just discuss the overview, and then we'll move on to steps one by one, the reactions one by one. Okay. First of all, let's discuss the overview. Okay. If you see you see these rounded these things, right? So these rounded things are basically simply telling you that how many carbons are present over here, right? So don't get fascinated to why these rounded things, they're not proteins, they are simply just telling you the carbon skeleton, right? How many carbon are present in each compound. Okay, for example, glucose has 1, 2, 3, 4, 5, 6 carbons, right? So it's telling you about the carbon skeleton over here. Okay, so glycolysis, remember that glycolysis occurs in two phases: phase one, phase two. What is the phase one? Phase one is a preparatory phase. What is a preparatory phase? Wherein we are using the energy. If you see here, we are not, ATPs are not being formed. Okay, ATP is being used here. ATP is being used here, right? And then ADP is released. So in this preparatory phase, you have to remember that no energy will be produced. So energy is consumed. Very important point. This is sometimes, we do have, we do prepare questions. Okay, we do have questions based on this energy preparation phase. Okay, so you should be thorough with this. Okay, now you will see that we take glucose. Let's start with the step one. Okay, altogether we have 10 steps in the glycolysis. Step one to step five is the preparatory phase, or the energy investment. We invest the energy over here. So this all is the preparatory phase. Step one, what do? We have a glucose. How did we get that glucose? We ate food. Food is converted into disaccharides, then finally monosaccharides, and monosaccharide was absorbed by the cell, and now cell is going to do glycolysis. It was transported into the cytoplasm of the cell, and now we have the glucose. So simply glucose will be converted into the glucose 6 phosphate. As you can simply see what is happening here, you know, the first three steps of glycolysis are the energy activation step. Sorry, not energy, sugar activation step. We will activate this sugar. Let's see how. Okay, so simply glucose will be converted into glucose 6 phosphate. As you can see here, one phosphate was added here. And where did we get that phosphate from? From the ATP. What was the enzyme? Hexokinase. You have to remember the substrates, you have to remember the products, you have to remember this, because the whole chapter is based on all this. So you will not email me anybody and do we need to know the reactions? Yes, you need to know the substrates, you need to know the enzymes, you need to know the products, you need to know the how many ATPs are produced, how many are used. So please remember that, right? So there's no question about not memorizing this stuff. This is very important. You have to. Okay. So once we have the hexokinase, okay, now we have the glucose 6-phosphate. Okay. So simply we use the energy over here. The phosphorylation took place. That is all. Energy was used here. Okay. Or you can say glucose was activated here. And how did we activate the glucose? By using the phosphate from the ATP. Okay. Once you have the glucose 6 phosphate, it will be converted into the fructose 6 phosphate. Okay. So can anyone tell me what is happening here? If you see here, we had glucose earlier and now we have the fructose. So what did we do? Which reaction did we do? We simply did the isomerization, right? So how we did the isomerization, we call it a tautomerism. We'll talk about that in a while once we discuss the reactions. Let's try to discuss the overview first and then we move on to the reactions one by one. Okay. And the enzyme which we used here was the phosphohexose isomerase. So we had an aldehyde first and now we have the ketone sugar here, right? So remember the enzyme. Now after fructose 6 phosphate, once we have fructose 6 phosphate, then we have the fructose 1,6 diphosphate. Again, when it's di means two phosphates were added. Where did we get the other phosphate from? Again, we use the energy here. So it's your responsibility to remember which steps, right, uses the energy. Okay, so if I give you the reaction steps, so you should be able to know that when we convert glucose into glucose 6 phosphate, we are using ATPs. You have to memorize that when we convert fructose 6 phosphate to fructose 1,6 diphosphate, we are using the energy over here, right? So you need to remember all this. So ATPs are used here, energy is used here. Okay. So step 3 was simply again using ATP, and it was again a second phosphorylation step here. One was here and one was here. So this is the second phosphorylation step over here. Once we have fructose 1,6 phosphate, and then we call it a splitting of the sugar. As you can see here, I had 1, 2, 3, 4, 5, 6 sugar molecules over here. I mean, 6 carbon atoms over here, right? So in this step, right, the splitting of the sugar will take place. What is the splitting of the sugar? The cleavage of your fructose or the splitting of your fructose 1,6 diphosphate will take place so that it can form the trioses. You know what are trioses? Carbohydrate chapter. Remember, dihydroxyacetone, we did this. Dihydroxyacetone and the glyceraldehyde 3-phosphate. We did this in carbohydrate chapter, right? So your C6 carbon compound is converted or it's cleaved, it's broken, okay? It's cleaved into two fragments, okay? One fragment is dihydroxyacetone phosphate and second one is the glyceraldehyde 3-phosphate. Remember we discussed it, right, in carbohydrates chapter, okay? The triose sugars. Okay, and these are interconvertible. When I say interconvertible, what does that mean? Okay, that they are two isomers, aren't they? Yes or no? Everybody, are we all awake? Okay, so here, which is the enzyme which we have here? We have the phosphotriose isomerase. Okay, or sometimes we simply call it the triose phosphate isomerase. Okay, or phosphotriose isomerase or triose phosphoisomerase. Okay, now after this, okay, once we have the glyceraldehyde 3-phosphate, right? So this was the first step, which was the preparatory phase, step 1 to step 5, right? It was the preparatory phase. Now, glycolysis is divided into another phase called as a phase 2, right? In phase 2, you will see that this glyceraldehyde 3 phosphate, this can also be converted into one more molecule of glyceraldehyde 3 phosphate. So you will have two molecules here, right? So that is why, as you can see here, everywhere you will be using the word 2, 2, 2, 2 after this. Remember this point. This is a very important point. Okay, we'll discuss it again in the reactions. Okay, so now these six steps are called as a payoff phase. When I say payoff phase, what does that mean? That means that in these next six steps, okay, we are going to produce the energy. As you can see here, we produced two ATPs here, and we produced two more ATPs here, and we also produced NADH here, which can be used later on. Okay, is it clear to everyone? In what are we going to do in these next six steps? We are going to produce the energy. Okay, and as you see here, now we are not working with six atoms of carbon. We're just working with the trio sugar. We're working with three atoms of carbon, right? So that is why, what do we have after glyceraldehyde 3 phosphate? It is converted into 1,3 bisphosphoglycerate. Okay, 1,3 bisphosphoglycerate. Is it clear? And then what is happening over here? Anybody? We have this is an oxidation step over here. Okay, well, as I said, that we'll discuss here in detail about the reactions. Okay, once we have the structures also there. Yes, Amjad, I will repeat. This is called a payoff. Payoff means getting something from someone. Oh, okay, so here we are forming the ATPs, right? So what is this these what are these steps going to pay us? These steps are going to pay us ATPs, right? So this is ATP formation steps, right? So these all steps, okay, rather there are not, all the steps are not ATP formation steps, but as you can see here, this is an ATP formation step and this is an ATP formation step, right? But in generally, the second phase is called as the payoff phase. Okay, everybody, did you all understand? Okay, so as you will see here, once we have the glyceraldehyde 3 phosphate, it will be converted into the what, it will be converted into 1,3 bisphosphoglycerate. Okay, you will see that here it will be dehydrogenated. So hydrogen will be removed here, and those hydrogens will be given to NAD positive, which will turn into NADH over here, right? So you will see that once we discuss this step, you will see that we'll have not only dehydrogenation, okay, we will also have phosphorylation. That is why we have 1,3 bisphosphate, right? We also added a phosphate over here. Look at this, we added a phosphate over here, right? So the enzyme is the dehydrogenase is the enzyme over here. Okay, and once we have, now we have 1,3 bisphosphoglycerate, which is again what, a 3 carbon compound. Okay, after that we have what, the step 7, we will produce the 3 phosphoglycerate is produced. Okay, from 1,3 bisphosphoglycerate, 3 phosphoglycerate is produced, and you have to remember the enzymes: phosphoglycerate kinase enzyme. Okay, so what are we going to do in this step? In this step, you will see that once we discuss, you'll simply see that phosphate molecule will be transferred, okay, from one part to the other part, and then we have the 3 phosphoglycerate, and this is the step where ATP is produced. So step 7, ATP is produced, and that phosphate will be given to the ADP, and which will result in the formation of ATP. Okay, so simply transferring the phosphate to the ADP so that we can produce the ATP. Okay. Once we have 3-phosphoglycerate, and then we will have, it will be converted into 2-phosphoglycerate. Okay, and the 2-phosphoglycerate, what is happening here, we'll see that here the phosphate linkage will be moved from one carbon to the other carbon, which we will see. I will show you that in the reaction step, how the phosphate linkage is moved from one carbon to the other carbon. Okay, from position number 3 to position number 2, how it is moved. Okay, so that would be step A. So no energy is generated over here. So after that, once we have the 2-phosphoglycerate, and at the end we'll have the phosphoenol pyruvate. We also call it sometimes PEP. Okay, so phosphoenol pyruvate is produced, also called, we call it, PEP. Sometimes in short form we write it like this, PEP. Okay, and here simply the water will come out here, and the enzyme is the enolase. No ATP is released here. But then the last step, where the phosphoenol pyruvate is, finally we have the two molecules of pyruvate over here with the production of ATPs. Two ATPs also produced. We call it the pyruvate kinase over here, right? And you will see that this pyruvate kinase is such an important step over here, that you will see that it plays a very, very important role in the regulation of the glycolysis. Very important. If pyruvate kinase is, because once we have pyruvate kinase, what inhibits, what is inhibition now, inhibition of enzymes now? Is it the second phase called as a payoff, or the second phase, Mustafa? The whole phase, this whole phase is called as the payoff, or the energy generation phase. Okay, and this whole phase is called as the preparatory phase. Preparatory phase. Preparatory phase. Okay, Mustafa, is it clear? And the first three steps, 1, 2, 3, these three steps are called as, anybody? I said that when we started it. What are the first three steps called as? What are we doing in the first three steps? We are activating the sugar, right? Isn't phosphate going? Yes, guys, I'm going to discuss that in a while, right? So here, this is called as the sugar activation. The first three steps are the sugar activation. Is it clear to everyone? So what did we produce at the end? At the end, we had two molecules of the pyruvate. This is the our end product. We wanted this. This is still a high energy compound. But I wanted to tell you about the pyruvate kinase enzyme. This enzyme plays a very, very important role, right? It is inhibited by the ATP. Yes, we learned that in the previous chapter. It's inhibited by the ATP. What does that mean? That means if you have, a lot of ATPs around in the cell, do we need glycolysis at that time, guys? If there are a lot of ATPs in the cell, do we need to produce energy at that time? No, of course not. Very good. So what causes the inhibition of the enzyme? The pyruvate kinase is inhibited if we have plenty of ATPs in the cell, so that glycolysis will not take place. So that is why I said that pyruvate kinase is a very, very important enzyme. And I think we learned that previously, which chapter was that? I don't remember which chapter was that, but we learned that previously, right? And then pyruvate kinase is activated by the, if we have what, if there is no ATP, let's say in the cell we have everywhere AMP. What is AMP? Adenosine monophosphate. Is it a high energy compound, AMP, guys? Do we have any anhydride bond here? See, this is the time when you have to collaborate everything. Remember, ATP has two anhydride bonds, but AMP has no, isn't it? So when you have a lot of AMP present, that will activate this enzyme. So, you should always collaborate and, try to connect it with the previous chapters, the enzyme inhibition and the activation. So inhibition by AMP and activation by sorry, inhibition by ATP and activation by AMP of this pyruvate kinase enzyme. Okay, so the moment you have a lot of ATP in the cell, glycolysis is shut down, right? The moment we have less ATP in the cell, right, and we have a lot of AMP in the cell, at that time glycolysis pathway will be speeded up, or it will take place in the cell, right? This is that's why I said that this enzyme is very, very important, pyruvate kinase enzyme. Did we all understand the basics of it? Are we all okay? I mean, the overview, then we will go one by one with the reactions. So this is like, this table is in your book, so you need to know this table, okay, this flow chart rather, okay, and then we need to know what is happening in each reaction. uh, I think somebody emailed me about the reaction steps. See, ladies and boys, you have to remember that each and every reaction step is important, only the structures are not important. I'm not going to ask you about the structures, to draw the structures over there, but you are expected to know what happens in each and every step. Okay? Are we okay about it? Did you all understand? I think somebody had emailed me about it. I don't remember who emailed me. I wanted to reply back, but then I wanted to discuss this in the class and then reply back. Okay? So, yes, reactions are important because reactions tell you what is happening there. But then, because the entire chapter is based on these reactions, right? But then, yeah, of course, you're not expected to know what is the structure of it and how did it take place. No. Just remember the way I explain you. This is phosphorylated, what enzyme we use, except the structures. The rest you have to know about it. Okay, so let's start discussing the 10 steps of the glycolysis. So I told you the first 5 steps are the preparatory phase where we are investing the energy, where we are giving the energy, right? And then I told you the first 3 phases are what? The sugar activation. So what is happening here? We have glucose, alpha D glucose, okay? It will be activated to form the alpha D glucose 6 phosphate in presence of the enzyme called as the hexose kinase. Okay, look at this. Excuse me. We had ATP here, and at the end, what did we get? We get ADP, right? So we use this phosphate from the ATP to phosphorylate the glucose. That is all we did here. We simply did the phosphorylation step, nothing much about it, about this step. Okay, so if the question says that how is glucose activated, you will say that glucose is activated by the addition of phosphate. Where did you get that phosphate? We got that phosphate from the ATP. Phase 2. Phase 2 is simply the isomerization. Okay, what is isomerization? You know that glucose and fructose, right? One is an aldehyde, one is a ketone, isn't it? Remember the structure? What was the structure called? All these things you have to go back, and all these things will be there in the final exam.
Pair conformations.
Yes. Okay, what about the fructose? What was this called?
Haworth projections.
Very good. Haworth projections, right? Are wonderful, right? The cyclic structures of your carbohydrates. Yes, Mustafa. Mustafa, the first step is very easy. In the first step, we simply had a phosphate here, which was added to the glucose at this point, right? And it formed the glucose 6 phosphate. That's it. So energy was used here from the ATP, and then at the end we had ADP produced. Okay, one phosphate bond was used over here. So it's called as a phosphorylation of the glucose or the alpha D glucose. Is it clear, Mustafa? Simply the phosphorylation step over here. Okay, now second step is the isomerization step. What is the isomerization step? In the isomerization step, simply you have to remember that what is happening here, we have glucose 6 phosphate, okay, and in presence of the phosphohexose isomerase, we result in the formation of the ketose. So we know that glucose is an aldehyde and fructose is a ketone. So simply this structure will be opened, right? If you remember, we spoke about the furanose rings and the pyranose rings, right? So this structure will be opened, the glucose 6 phosphate, and as we open the glucose, right, we open this the hemiacetal ring, right? If you remember, we spoke about they cyclize this is from the carbohydrate chapter, right? So this is an open structure of the glucose. So you will see that glucose will simply glucose 6 phosphate will open, okay, and as its ring form will open, what's going to happen? It's going to be converted into, first of all, an in indiol, okay, and which is basically a cis form of it, and then finally it is converted into another isomer called as the fructose 6 phosphate. Okay, it first produces an open chain fructose, right, carbohydrate chapter. Okay, once we have an open chain fructose, okay, I don't know whether you remember it or not, in carbohydrate chapter we saw that what does it form then? It forms the, again, the ring structure, because glucose, fructose, they all enjoy, right, pentoses and hexoses, they all enjoy the cyclic structures, isn't it? We spoke about that it's the same thing, right? So they enjoy to stay in the cyclic structure rather than in the open chain structure, right? So you don't have to go into detail. We usually call it a keto-enol tautomerization, but you don't have to remember all that. You simply have to remember that one form of the isomer is converted into another form. So aldehyde is converted into the ketone form. That is all you need to remember here. Okay, but remember the rings will open first, as you can see over here, and then they will cyclize once again. This fructose 6 phosphate will again cyclize, and as you can see here, it has a cyclization structure again here. Okay, so simply it is an interconversion step, reorganization step, that is all. Okay, nothing more is happening in this step. So do we have any question about step number 2, boys and girls? So simply remember, it's an opening up of the glucose hemiacetal ring to its open chain structure, and then finally cyclizing once again. That is it. Into hemiketal form. Isn't this hemiketal? This is hemiacetal. This is hemiacetal because it has an aldehyde, and this is the hemiketal. Remember that. So your hemiacetal is converted into the hemiketal form, right? This is what's happening here. This is an isomerization step. Nothing else is happening here. Step 2. So step 3, what do we have? Now we have the hemiketal now, not the glucose. We have the alpha D fructose 6 phosphate here. Okay. And what is happening here? Again, we are using energy of the ATP here. Again, this step is an energy utilization step. Okay. Again, we are doing activation of the sugar here. We are activating fructose 6 phosphate into fructose 1,6 bisphosphate. Look at this. Our phosphoric acid at position number 1 and position number 2, which results in the formation of fructose 1,6 bisphosphate. Remember, it's just a phosphorylation step where we use the phosphate from the ATP to phosphorylate the fructose 6 phosphate. Are we okay about 3 steps? Any question till now about the 3 steps? Okay. Now, if you remember, what did I say? Mohammed, bisphosphate means 2-phosphate not closed. No, Mohammed. There's a difference between diphosphate and bisphosphate. Diphosphate means when you have 2 phosphate groups, okay? But when we use the word bisphosphate, bis means 2 phosphoric acid, Mohammed. Okay, so remember this is the difference between the, if I say 1,2 diphosphate Or if I say 1 to bisphosphate. Bis means when you have the phosphoric acid. Okay, not the phosphate, as you can see here. Okay. Then comes your reaction 4. Reaction 4 is very critical because if you remember, I showed you here in reaction 4, there will be what? There will be cleavage, breakdown here. Okay. So, what is the cleavage? The cleavage is your fructose 1,6 diphosphate is going to get converted into what? Is going to get converted into the triose sugar. One is the dihydroxyacetone phosphate, another is the glyceraldehyde 3 phosphate. Okay. So actually if you go back to this step here, what is happening in step 4. If you see here, we had fructose 1,6 biphosphate, right? This one. Okay, if you see here, what is happening simply, we have an oxidation happening over here. Okay, so there will be an intermediate carboxylic acid formed here. Okay, but then that will be phosphorylated later on. Okay, that we'll see later on. Okay, that is in the next step happening. Okay, but here you will see that it will simply split over here. Okay, and form the what? And it will form uh, the dihydroxyacetone phosphate and the D-glyceraldehyde 3 phosphate over here. Okay, so in short, you will have to remember in this step, one molecule of this fructose 1,6 bisphosphate, which had 6 carbons, if you can count here, which had 6 carbons over here, it was cleaved, broken, right? It was broken into two molecules of 3 carbon unit each. That's all you need to remember over here, right? And the enzyme here was the aldotase. Okay, that's all you need to. So next step you will see that. Are we okay till now? Yes. Now you will see that in the next step, what's going to happen here? Okay, okay. In the step 5, what you will see here, what is happening here? So it's just the isomerization step here. What is isomerization step here? So you have the dihydroxy acetone phosphate. It will be isomerized into what? To form the D-glyceraldehyde 3 phosphate. Okay, and enzyme here we have the, because it's a triose sugar, right? So it's the phosphotriose because isomerization is happening here. So it's the phosphotriose isomerase is the enzyme here. Okay, is it clear? So what is the net result now? We started with one molecule of glucose. Okay, it is now cleaved into two molecules of glyceraldehyde 3 phosphate, right? So this step and this, actually this step is called a splitting step, and this is isomerization, but sometimes we call these two steps as splitting steps. So remember, this is a very important point. That is why if you see here, we are using the word two after this. Okay, everywhere we are using two times. Okay, because what was happened? Because dihydroxyacetone will be isomerized to its isomer, called as glyceraldehyde 3 phosphate by the phosphotriose isomerase. So remember, that is why after this we'll be using the word 2 everywhere. So if I say 1 ATP is produced, you have to eventually multiply it with the 2. Did we all understand this step? Yes, ladies, are we okay till now? The first 5 steps were what? Were the energy utilization steps, isn't it? We use the energy there, the preparatory phase. Now let's discuss the last 5 steps where the energy is produced. So I was telling you that now we have two molecules of what? Glyceraldehyde 3 phosphate. What's going to happen here? Okay, so you will see that glyceraldehyde 3 phosphate, okay, it is dehydrogenated. Okay, it is dehydrogenated and at the same time phosphorylated also. So two things are happening here. One, the dehydrogenation, and number two, phosphorylation here. Okay, both the things are happening so that we have 1,3-bisphosphoglycerate. And the hydrogens which we removed from here will be used to convert NAD positive to the NADH. Now we have the reduced form of NADH, which we can use later on to generate the ATPs. So remember, very important point, at which step does the NAD positive is reduced to the NADH? It is at the step number 6 when we have glyceraldehyde 3 phosphate converted into 1,3-bisphosphoglycerate. So these kind of questions you might expect. Is it clear till now? Mohammed, I will talk about what are the reversible steps and what are the irreversible steps, right? So usually this step isn't, the all the steps are reversible step except the three steps. We'll talk about that. Step 1, 3, and 10 are irreversible. Rest, you will see that most of the steps are reversible in the glycolysis, Mohammed, because we did not take the chapter, the last chapter at the end, okay, wherein we will synthesize the glucose, gluconeogenesis. We are not taking that chapter. There you will see that most of the steps of glycolysis are reversible, right? All of them are reversible, right? You're right, Mohammed. It can go back to the NAD positive, okay, but except the three steps which are irreversible, which I will be discussing at the end, right? And or let me show it to you here. Let me erase this. Mohammed, please pay attention. All the steps of the glycolysis are reversible, right? Except step 1. This one is non-reversible. Okay, this hexokinase one. Okay, and then we have the step number 3. This one is also what? Irreversible. Okay, where we have the enzyme phosphofructokinase. Okay, and then the last step, this one, let me erase this first. Okay, the 10th step, this one, right? Except these steps, all the steps of the glycolysis are reversible. Muhammad, did I answer your question? Okay. So where were we? Okay, good, alhamdulillah. So we were here. Okay, in step 6, guys, what happened in step number 6? In step number 6 we simply removed the hydrogens, gave it to NAD positive, right? What happens? These hydrogens were picked up by NAD positive and it formed the NADH. So in this way, what did we do? Some of the energy we transmitted to the NADH, which we can use later on to produce more ATPs. So that is why I said that this, from step 6 to step 10, all these steps are all the steps are called as the payoff phase. Even though only ATPs are produced at only two places, but this is also going to pay us ATPs later on, right? Because we have stored some energy in the NADH. Are we okay till now? Step number 6 is clear to everyone. Shall I move to step number 7? So what is happening here? Two things, dehydrogenation and phosphorylation is happening here. Now, step number 7. Step number 7, what did we have? We had 1,3-bis phosphoglycerate here. Okay. Simply 1,3-bis phosphoglycerate will form two molecules. Remember, okay, it will form what? Two molecules of 3-phosphoglycerate. What is happening in step number 7? Can anyone tell me?
Transfer of phosphate group.
Yes. See, we have this phosphate group, right? We will give this phosphate group to the ADP, and this is an energy generation step. So that's how we produced the two molecules of ATP over here. Very important point here, right? So this is a payoff step, right? Where we produce the two molecules of ATP over here. Okay. And if you remember, I told you that this is called as the substrate level phosphorylation. Okay. Where the phosphate donor, who is the phosphate donor here? Who donated the phosphate to ADP? Who was the one who donated phosphate to the ADP? It was the, wasn't it the substrate? Isn't this the substrate? Yes. So it was the substrate over here. It was the 1,3-bisphosphoglycerate. So this also you need to remember, right? So why is it called as the what? Substrate level phosphorylation. Okay, because the ATP which is produced here, so this is the first level, first substrate level phosphorylation. Second will be coming up in a while, the last step. Okay, where the two ADPs will be converted into two ATPs. Okay, then we'll move on. Now what do we have? Okay, we have the 3-phospho is okay. Is this step clear to everyone? So we simply did for transferring the phosphate group, okay, from 1,3-bisphosphate to the ADP. That is all we did. Nothing much about this. Okay. And now in step number 8, what are we doing in step number 8? Step number 8 is simply again the isomerization step. Okay. So isomerization of 3-phosphoglycerate to 2-phosphoglycerate. What did I do here? Okay, the phosphate linkage, okay, which was where, can you imagine what is happening here, which was at 1 2 3 carbon atom, right? I simply transferred it to the 2 carbon atom. So that is all it happened. So it resulted in the formation of an isomer. Okay, so this phosphate was moved from carbon number 3 to carbon number 2. Okay, and we have 2-phosphoglycerate formed. So nothing much happening in step number 8. And then step number 9. What is step number 9? Step number 9 is dehydration of the 2-phosphoglycerate. What is happening? Dehydration here. Okay, what is coming out? Water is coming out. Do you see here? And as a result, this double bond was formed over here, right? In presence of the enzyme called as the enolase here, right? So simply the water molecule came out and we have a double bond here. So we formed phosphoenol pyruvate. And then we are just one step away in forming our final product, which is the pyruvate. So phosphoenol pyruvate will finally be converted into to form the what? To form the pyruvate, and the enzyme will be the pyruvate kinase. Okay, again a phosphate from the phosphoenol pyruvate will be given to what? Will be given to the ADP, which will result in the formation of two molecules of ATP. So again, this is the second substrate level phosphorylation. We took the phosphate from the substrate and gave it to the ADP to form the ATP. So that is why this is the second level, second substrate level phosphorylation. So you should know these steps, especially the steps which produces ATP, and especially the steps which produces NADH, and especially the steps which uses NADH, which uses ATP, right? So with that, we have the pyruvate now at the end. And if you remember, I told you this enzyme is very, very important, right? So if the question says that which of the following enzymes play an important role in the regulation of glycolysis, you have to say it's the pyruvate kinases, because it is inhibited by the ATP and it's activated by the AMP in the cell, or ADP in the cell. Now tell me, do we have any question? Everybody understood it here? There's one video also here. I will upload that video on the Moodle. Okay. Now this is the overall. So overall reactions are also again very important. In the overall reaction, what did we produce? We produced what? 2 NADH. We actually produced 4 ATPs. Remember, we produced 2 ATPs here, right? And then we also produced 2 ATPs here, right? So actually we produced 4 ATPs, in the glycolysis, but out of 4 ATPs, if you remember, in the first 5 phases, we used the ATP twice, right? We used it once here and once here. So that is why the net result is only 2 ATPs and 2 NADH are produced at the end of the glycolysis. Glycolysis is pretty much easier than the citric acid cycle. Do you agree or no? You don't? Yes, guys and ladies? It's easier than, the citric acid cycle. It's a little simpler, yes. You need to remember a few things. Now, a very important point. When you produced your, once you have your pyruvate, what are you going to do with it? Now, please pay attention, everyone. Okay. Now, once we have the pyruvate, there are many things which can happen. Okay. Once we have the pyruvate, okay, there are many things. One, if the oxygen is available, we'll talk about that in a while. If oxygen is available, first talk about that. If oxygen is not available, what are we going to do? If there's no oxygen present, what are we going to do? Right? Can anyone tell me? See, you have to remember that glycolysis, usually I've seen students saying that, oh, glycolysis is an anaerobic process. You have to remember, it depends upon what is the condition of the body. Okay. So it depends upon whether oxygen is available or if oxygen is not available. There are two things which can happen. Okay. Once we have pyruvate produced, now we say fate of pyruvate. What is fate of pyruvate? Pyruvate has two fates. Like it can either go, if a lot of oxygen is available, a lot of O2 is available, okay, it can complete the other steps of the what cellular respiration. What are the other steps? This pyruvate will be converted into acetyl CoA, and then acetyl CoA will enter into Krebs cycle, and then the electron transport chain. So like a lot of ATPs will be produced, right? So this is like the normal thing which will happen. So all 4 stages are completed if oxygen is available. But what if oxygen is not available? What do you think? Can you tell me any scenario when oxygen is not available in the body? So where can it happen? We did that last semester in muscular system, I think. Nobody? When is the condition when body has no oxygen? Heavy exercise, absolute, it will produce a lactic acid, absolutely, Mohammed Tashir, both of you correct, right? So the condition when we have less oxygen available is that when you're vigorously working out, your muscles are constantly working out, right? At that time, what will happen that the pyruvate which is produced can be converted into the lactic acid. Okay, we call it the anaerobic glycolysis at that time. So at that time, the remaining steps of the cellular respiration will not occur. Okay, and ATPs will be produced by what? ATPs will be produced by the conversion of pyruvate into the lactate. Okay, and this happens whenever you're intensely working out, when there is less oxygen available, because if you remember, we said that when oxygen is available, it's a very long process. At that time, your muscles cannot wait for so many steps. It instantly wants ATPs to be produced, because this step is going to give it how many ATPs? Two molecules of ATPs will be produced. But what is the advantage of this step? It happens more quickly with very less time. And it happens, remember the advantages last semester? It happens even if oxygen is not available. There are not many steps in converting pyruvate to the lactate. Okay, so in absence of the oxygen, pyruvate is converted into lactic acid actually. Okay, then lactic acid will give you lactate. Okay, since lactic acid is converted into lactate, so you will see that lot of H positive ions are available, right, which will give the muscle soreness. We discussed that last semester. The muscles will start paining. Okay, so once you will stop working out, you stop doing exercise, what will happen to this lactate, guys? Did you ever think about it? Once you will stop working out, you are like excessively tired. You're so tired that your muscles will say that no, I cannot work out more because I have plenty of lactic acid in the muscles. I have plenty of H positive in the muscles. So your pyruvate will not be ready to be converted into lactate. You have exhausted yourself to such an extent. You're exhausted so much that no further pyruvate can be converted into lactate. So what will happen to the lactate which is already available? Go out of the muscle tissue. Very good. And then what will happen? Very good. Very good. It will be converted back to the glucose. Absolutely. Do you know that? Once you are tired to such an extent, as I said, that why do you give up? Even if your mind says that I really want to work out more, but your muscles say that your muscles are like, no, we cannot, we can't go beyond this, because at that time there's an immense accumulation of lactate or lactic acid or H positive ions in the muscle. No more conversion of pyruvate into the lactate will take place. But then slowly and slowly, as you rest, as you settle down, you start taking more and more oxygen. More and more oxygen becomes available, and this lactate, as more and more O2 is available now, this lactate is taken to the liver. Okay, it's taken to the liver, right? And this lactate will be slowly and slowly converted back to the glucose in the liver. Okay, and that process is called the Cori cycle, CORY. You will learn that in biochemistry in College of Medicine. Here you don't have that. Yeah, acidosis. Yes, Tanya, acidosis because of the hydrogen ions. Okay, so here this lactate will be will be taken to the liver. From the liver, it's converted into the glucose. Okay, and then glucose is released from the liver. See, one more function of the liver. I told you liver has almost 300 functions. One more function of the liver will convert this extra lactate into the glucose, right? And then this glucose will be released from the liver. It will be transported to the cells. Okay, and the cells can again use that glucose for the energy. So this conversion of lactate into glucose is called as the Cori cycle, which you will learn in biochemistry, but in College of Medicine, not here, right? Okay, so we produced two molecules of lactate and two molecules of ATP at the end. Okay, now let's say a lot of a lot of oxygen is available. Let's say you're not doing any vigorous exercise, right? So when a lot of oxygen is available, what is going to happen? This simply pyruvate is converted into, as I told you, it's converted into acetyl CoA. And this acetyl CoA will later on enter into the TCA cycle. But the question is, how is this, okay, how is this pyruvate converted into the acetyl CoA? That is the question, right? So we call this phase as a transition phase, or we call this phase as a formation of an acetyl CoA phase. Okay, so this is our aim. If a lot of oxygen is available at that time, your pyruvate, this pyruvate, okay, this pyruvate will be converted into acetyl CoA by simply, first of all, decarboxylation. As you see here, this carbon dioxide is what? Lost, or this, the pyruvate will lose this carbon dioxide, right? And this is this carbon dioxide which is produced over here. Okay, so the carbon dioxide which you produced from your mouth came, the first time when it was produced, came out from the pyruvate. So this is the first time when the carbon dioxide was released. Can anyone tell me, when did we release the carbon dioxide second time? When do we release carbon dioxide? In which cycle? Other than here, where else do we release the carbon dioxide? You can expect these kind of questions in the exam. So this is the place where you release carbon dioxide for the first time. And when do we release it for the second time? TCA cycle, absolutely. So the very good guide. So the two places, so the two cycles where we release the carbon dioxide, one is when you convert pyruvate into acetyl CoA for the first time, and second time in the TCA cycle or the Krebs cycle. Okay. So what happens? So here we pyruvate, so carbon dioxide is removed. Actually, many steps will happen over here. First of all, the carboxylation, okay, and then even the oxidation will take place. Okay, so you will see that the hydrogen atoms will be removed from here. It will be given to the NADH. That is why we were able to produce the NAD positive. The hydrogen atoms will be given to NAD positive, and that is how we produce the NADH also. So all these things are very important when it comes to energetics. When do we produce NADH? You have to remember each and every step. When did we produce ATP? When did we produce NADH? Very, very important. So if you see here, we have produced one more NADH here, which can be used later on to produce the ATPs. Okay, and that's how we produce the acetyl CoA. So what did we use here? What is this one? CoASH. Guys, what is this CoASH? What did we use in this reaction? Yes, boys and girls, which enzyme is used here? What is CoASH? Yes, Guide, Tashil, Mohammed, Tanya. Nobody?
Coenzyme A.
Coenzyme A, guys. Of course, we use, very important, we use the coenzyme A here, right? So, so remember, we call it coenzyme A. Actually, the SH is just to show here that we have a thiol group with the coenzyme A. So coenzyme A, remember, there was a question which we had put, I think, last year. What is the other name of coenzyme A? We also call it coenzyme ASH to show that it has a thiol group, right? If you remember, I told you last time, things can, this is like a hanger. If you remember, I told you that. Okay, so this acetyl group will combine with this coenzyme A. That is how we got the acetyl CoA at the end. Okay, so this was basically how we converted pyruvate into acetyl CoA by reactions like one, decarboxylation, and two, oxidation. So that is why together it's called as oxidative decarboxylation. Okay, and we form the acetyl CoA, which can enter into the TCA cycle, right? So very important, you should be knowing what happens under aerobic condition and what happens under anaerobic condition. Very important, right? Both the things we discussed here, aerobic as well as the anaerobic. But then they've also given you how the ethanol is also produced. So you can just have a look about this reaction, how ethanol is produced. Okay, so we can convert the pyruvate, okay, in the yeast. Okay, yeast can, yeast are also microorganisms. We use yeast in ripening of the bread, isn't it? So or in ethanol making. So these yeast, okay, they have to regenerate NAD positive. What do? Okay, so they convert the pyruvate first of all into acetaldehyde. Okay, and once in presence of the pyruvate decarboxylase, where the carbon dioxide is released. Okay, and then this acetaldehyde is further reduced to the ethanol, as you can see here. Okay, and we used NADH here. So remember the enzyme called as alcohol dehydrogenase. Yes, there are many other reactions. You're right, Tanya. You're right? But there are many other reactions also happening there. Okay, so this is how yeast produce the ethanol. This is the formation of the ethanol. So this is just one slide over here which you need to remember. Okay, and other than that, what else here? Yeah, and other than that, how do we regenerate NAD positive? See, the glycolysis needs NAD positive, right? And if oxygen is available, if the cell is doing cellular respiration, okay, the NAD positive, ladies, please pay attention. One last minute. See, we are using NAD positive here. This NAD positive. How did we get this NAD positive? If you go back to cellular respiration, remember when we discussed at the end, when we discussed the oxidative phosphorylation last class, when FADH2 was converted into FADH positive, NADH was converted into NAD positive. Remember last class, ladies? The electron transport chain? Yes or no? Boys, girls, where are you? Everyone? Is it difficult? Is it difficult to comprehend? Yes, Tashil. Yes, Tanya, what to do there? But we are just about to finish. We are about to finish. The class is about to get over, right? So I just wanted to let you know that if you remember, if you go see, that's why I say that everything is interconnected. If you would have revised your last lecture, you would have understood how do we get the NAD positive. In last class, when we did the last part of it, we said that NADH is converted into NAD positive, right? And it is this NAD positive which can be used in the glycolysis again, right? So if normally the respiration is taking place, all four steps of respiration is taking place, we can get this NAD positive from the electron transport chain at the end when it, okay, so your NADH is regenerated. But just in case if the aerobic respiration is not taking place, then how can we regenerate this NAD positive? At that time, the only source for us to regenerate this NAD positive will be doing what? We will go and do the anaerobic respiration, right? So if no oxygen is present, in order to reoxidize NADH to the NAD positive, we have to find a way out how we can regenerate this. And regeneration can be done by the anaerobic respiration. If you see here, we not only produce lactate here, but we also produce NAD positive here, which can again go back to the glycolysis cycle and can be used there to convert it into NADH. Yes. Okay. So that's all for today. So we just discussed about the glycolysis today, and then we discussed what are the end products of the glycolysis, and then we discussed what happens if a lot of oxygen is available. Okay, and then we discuss what happens if there's no oxygen which is available. We can form the lactate and the NAD positive is regenerated. But if oxygen is available, we can form acetyl CoA, which can go into the citric acid cycle and then continue the aerobic respiration. So in the next class, that is on Wednesday's class, we will discuss about the energetics. Okay, we'll first discuss about the pentose phosphate pathway, or we have a lot to discuss in the next class as well. Okay, then we'll discuss the energetics, and then we will move on to the other pathways, the beta oxidation of fatty acids. Okay, so anyways, this will be in the next class. So by that time, you all should revise as we are taking the chapters, because right now you have plenty of time. So please revise, ladies and boys, everyone. Okay, and on, as I said, that on in Wednesday's class we'll have a combined class wherein we'll finish the chapter first, and then later on we will discuss about the researches. And if you have any questions, you can ask me in the next class on Wednesday about your research reports. Okay, thank you everyone. Thank you everyone. Do you have any question? No. Okay, and when I have to join the next class. Thank you everyone. Bye bye. Yes, Mustafa, but not many days, but just couple of days, Mustafa. I'm going to extend it just for couple of days. Okay, because I really want to discuss on Wednesday. I want to give the input on Wednesday, and maybe from Thursday I can extend it to Saturday or Sunday. Saturday most probably, Mustafa. Yes, yes, I'm going to extend it. Okay, don't worry. Okay, thank you. Yes, Taulin, don't worry. I have all the emails. Okay, it's noted. Taulin, no problem there. Bye bye, everyone.